So you may have forgotten this, but I threatened at the beginning of this course that essentially everything we did would be in some way cumulative. That once we got through the exam on population genetics and natural selection and drift and so forth, that you weren't allowed to forget that stuff because it would come back up. Well, I'm afraid it is. One of the reasons that I had you watch that video by David Baum that showed kind of the branching or the evolving population of plants, kind of passing on genes from one individual to the next, and kind of scaling up to the population level, and then scaling up to the whole species level, and then scaling up to this huge macroevolutionary level, is because I wanted you to understand how the view that we're looking at now, this phylogenetic view over millions and millions of years, is really connected to the population genetic view that we've been using, that we used to look at things in the previous weeks of this course. You may have noticed that the diagram of all those plants evolving together, the population of plants exchanging alleles, looks very much like the diagrams that I showed you of the coalescent, of what happens when you have a population evolving and one allele is giving birth essentially to other alleles. And that's because they really are the same phenomenon happening at different scales. Okay, so what happens at the population genetic level eventually builds up to the point where it affects what's happening at the phylogenetic level. One of the ways in which this is important is with respect to a phenomenon called the molecular clock. Whenever you have a neutral sequence of DNA, over time in a population, it is going to be hit with mutations. And because there's no selection acting on that neutral sequence, those mutations are just going to accumulate. Drift is going to favor some of them. They'll just happen to drift to a high frequency. Others will happen to drift to extinction. But every so often, what will happen is one of those mutations will be fortunate and it will drift to fixation in a population. In fact, if you have many populations and they're sort of interbreeding over time, eventually some lucky mutation is going to drift to the point where it takes over the whole species. Okay, and now the entire species has a genetic sequence, the same genetic sequence that it didn't have before. Okay, this is happening all the time in all species all over the world. These mutations are rising, they're drifting, they're changing in frequency, and every once in a while, one of them will drift to fixation. That's what's called a substitution. A substitution occurs when a new mutation arises, and it drifts to the point where it's found in all the individuals in a species. Okay, an allele arises and it completely takes over a species. That's what a substitution is. Now, if you have two lineages, let's say two populations of the same species, for the most part, they're basically going to be identical at a neutral locus. So if you look at two individuals that are in two populations of the same species, for the most part, they're going to have the same genetic sequence no matter which individual you're looking at. It's going to look like this. Okay, here's the genetic sample from one population over here. Here's a genetic sample at the same locus from another population. Exact same sequences. Okay, they are 100% similar. But let's suppose that these aren't just any two populations. Let's suppose that this population is to the east of the Isthmus of Panama, just as volcanoes start forming in between North and South America, and this land bridge starts rising up out of the ocean. These say this sequence is in some species of, I don't know, some species of shrimp, which is found out in the Caribbean. This is the same species of shrimp that is found out in the Pacific. And now you've got this massive chain of volcanoes forming between them. These two populations are being cut off from one another. The Isthmus of Panama rises out of the sea, and these two populations will never meet again. What's going to happen to those two populations over time? Well, a mutation will pop up over here in the Caribbean population, and just by chance, it'll drift to fixation in this species, but not in this species, because these two populations are no longer exchanging genes, all right? We've got speciation underway. So whatever mutation, the mutations that happen in the Caribbean stay in the Caribbean. The mutations that happen in the Pacific stay in the Pacific. And the same is true for substitutions. Substitutions that happen in this population will stay over here. The ones that happen in this population will stay over here. And over time, what you're going to see is that these genetic sequences will drift apart. Okay, here are a couple of substitutions in the Caribbean that didn't happen in the Pacific, and vice versa, a couple in the Pacific. As more time passes, more of these will accumulate in both sequences. This genetic similarity between the shrimp over here in the Caribbean and the shrimp in the Pacific will erode. It's like the ticking of a clock. As time passes, more and more mutations happen in these two populations. They drift to fixation, and it happens regularly enough that you can basically go back and infer how long it's been since these two populations were separated by looking at the similarity in their genes. Okay, here's a different phylogeny of a lot of the vertebrates. We finally got away from that one phylogeny that you're probably sick of. And superimposed on this is a timeline. These are the times when we think these different lineages split away from one another. And this is going to be super obvious, but I'm going to say it anyway. When you have an organism like a kangaroo and an organism like a human, they started diverging from one another about 135 million years ago. That's not very long by evolutionary standards. And so what you would expect to see is that they have reasonably similar genes. You need to stop looking at that newspaper. There is a kangaroo in front of you. It might kick you. Oops, that wasn't supposed to happen at all. Let's move on. If you have a species that human beings diverged from a very long time ago, like say this shark and this human, that was back here in the Ordovician, I don't even know how long that was ago, about 400 million years, I guess. They're going to look very different in terms of genes. And so, of course, you can flip that around. If two species look very different in terms of their genes, they diverged from one another a long time ago. You can even get quantitative with this. This is the idea of the molecular clock. If you have a certain number of substitutions per base pair accumulating every X number of years, then over time you're going to see a certain amount of difference in their DNA. Specifically, if some number R is the number of substitutions per base pair that accumulate between two species every million years, then the fraction of differences in their DNA sequences should be 2RT, where T is the number of millions of years that since those species diverged. Okay, how do you know what R is? Well, if you've got lineages where you've got a good fossil record of when they split apart, you can look at their DNA. Okay, you know that for those species, let's say T is 50 million years, you know that their DNA is 12% different. Okay, so let's say we've got 100 million times R is 12. We have 12 by 100 is 0.12% per million years. That's R. Now that you've got R, you can use that to calculate T for all sorts of pairs of related lineages, whether you've got a fossil record for them or not. So all you need are a few points where you can calibrate your molecular clock, where you've got enough fossil evidence to calibrate your molecular clock, to get a good, precise estimate of R. And then you can use the molecular clock to determine when different pairs of lineages diverge from one another in the deep past. Okay, so in this case, what you've got is you've got a few samples where they've estimated the time since they split in millions of years from the fossil record. For this particular gene in this group of organisms, R is about 0.83 substitutions per million years. And then what you can do is you can look at the genetic difference between any of these different species that you've got, and you can estimate when they split apart. Okay, so for example, even without fossil evidence, we can look at the genetic similarities between a human and chimpanzee and infer that those lineages parted ways between 6.0 and 7.0 million years ago. If you want to know when the New World monkeys split away from the Old World monkeys, well, that probably happened between 37 and 52 million years ago, based on the molecular clock. This is great because there are a lot of groups of organisms that have lousy fossil records, and you can still get a decent reconstruction of their evolutionary history by using the molecular clock. Is it really this simple? Well, of course not. No. Nothing in this class is as simple as I'm telling you it is. Everything is an oversimplification. There are all kinds of complexities and hiccups to the molecular clock, most of which I'm not even going to talk about. One of the problems you have, though, I am going to talk about a couple of the problems. And one is that when you're looking at the fossil record, what you're getting is not a precise time that two lineages split apart. What you're usually going back and you're looking at is, okay, I know that there is something that looks like a horse, and there is something that looks like a dog, or a dog relative and a horse relative back here at this point in time. And so I know that they split apart at least 110 million years ago or whatever. But really, I mean, the estimate, you can have this really big range around the estimate. Okay? And so when you go to estimate R, there's a lot of uncertainty about what R really is. And so when you go to estimate other dates based on R, that uncertainty stays with you and it makes all of your estimates uncertain. Another fact that we know about, which is actually really interesting, is that the rate at which the clock ticks is not the same for different genes. It's also not always the same for different groups of organisms. Why it's different in different groups of organisms is very complicated, and there's a lot that isn't known there. We have a pretty good idea of why the clock doesn't tick at the same rate in different genes, and this should be something that you can guess based on your knowledge about the population genetic mechanisms that you've already learned about. Some parts of a gene are under more functional constraint. What that means is that for some genes, they're really important for the function of an organism, and certain parts of those genes are really important for the function of those organisms. And so when a non-synonymous mutation comes along and it changes that particular DNA sequence, it is very likely to be harmful. And so natural selection will weed it out. When natural selection weeds out a mutation, that mutation never becomes a substitution. Okay? So few mutations in really critical proteins and really critical parts of really critical proteins ever make it to be substitutions. And so they don't show up in the molecular clock. Okay? Other proteins where the sequence really doesn't matter very much, any mutation that comes along could become a substitution. And so those clocks tick fast. Molecules where there's a lot of functional constraint tick slow. Molecules where there's very little functional constraint tick fast. Here's an example of this. Cytochrome C is a very important gene. It's very important in metabolism. And so a lot of mutations that happen in this gene get smacked down by natural selection. When we compare different species in the present day then, we tend not to see very many differences between the genetic sequence of the cytochrome C in, say, fish and the genetic sequence of cytochrome C in, say, birds. The reason is that when you tweak that gene, bad things happen, and so it just doesn't get tweaked very much. In contrast, take the fibrinopeptides. Some of these peptides are actually, the sequence does not matter at all. It's just a piece of protein there taking up space. It could be any sequence of amino acids and it would do the same thing. And so any mutation to that sequence doesn't get thrown out by natural selection. This clock ticks a lot faster, and you can see it in the slope of this line. So when you calculate R for one gene, you can't use that estimate of R for a different gene because they might be under different functional constraint.